Hello again, and this is the last video about human reproductive system, and also it's about the growth and development in the embryologic period actually. Let me define what is growth. Growth is increase in size, and growth always occur with mitosis because mitosis increases the number of the cells without genetic variation. And human development is the process of growing to maturity. In biological terms, it entails the growth from one cell zygote to the adult human being. And cell differentiation is the specialization of cells in structure and function, because all cells comes from the zygote, and all the cells have the same genetic makeup with zygote. Morphogenesis is the process by which an animal takes shape and form. Cell division, growth, and differentiation are the three main events occur from zygote to fetus. Fertilization is the fusion of sperm and egg, we mentioned in the last video, and differentiation is from zygote to embryo. Some kind of differentiation occurs in the cells and the tissues and organs, and development to become the adult, the individual. Okay, development of human embryo. Let me explain fertilization from fertilization. Fertilization is occur in the oviduct when the sperm and egg is fuse their nucleus in the oviduct, and after this fertilization, immediately cell division mitosis start, and we call this event is cleavage period or segmentation. And this blastocyst, the sixth day, in the sixth day, the blastocyst implants to the endometrium of uterus, and we call this blastocyst, the shape of this embryo we call blastocyst. With the help of some kind of tropoblast cells, they have to form some kind of placenta in the future. Okay, this is the implantation. After blastocyst formation, the embryo implants into endometrium, and embryo, the blastocyst, release human choronoid gonadotropin, and this hormone is the hormone to detect pregnancy, used for detect pregnancy. And pregnancies can be terminated spontaneously due to chromosomal or developmental abnormalities. For example, ectopic pregnancy is one of them. When the fertilized egg begins to develop in the fallopian tube, it should be removed from this fallopian tube. And development is determined by zygote genomes and molecules in the egg cytoplasm called cytoplasmic determinants. Okay, after fertilization, embryologic development proceeds through cleavage, or we can call this period segmentation. Actually, the segmentation is the first week of embryologic state. After the segmentation, the blastocyst turn into blastula, or blastocyst in mammals actually turn into gastrula. We call this stage gastrulation, and three-layer gastrula form in the gastrulation stage. And at the end of this stage, organogenesis start, and from these three layers are endoderm, ectoderm, and mesoderm. All the organs are giving rise to the organs from these three germ layers. Okay, the first week we call this cleavage or segmentation. Start with zygote, and in this ends with blastocyst. It starts with zygote, ends with blastocyst, and when the zygote divide into first 2 cell, then 4 cell, each cell are called blastomeres. Each dividing cell, for example, this one of them is blastomere. And blastula is the ball of cell with filled field cavity. This one is the blastula, or we can call it blastocyst in mammals. It's filled field cavity, empty inside, and the empty part inside is called blastozoa. But before blastula, there is another structure we call this cell field wall called morula. Zygote, then morula, then blastula, or we can call it blastocyst. Each cell is called blastomere. The empty space is called blastozoa. And total mass of morula is equals to zygote, because cell divides without growth. The cytoplasmic amount doesn't change, but the increase of the number of the cell increase very rapidly. After blastocyst, there is a gastrulation start, and the gastrulation period start. These cells become endoderm, ectoderm, and mesoderm. 3 germ layers are formed in the gastrula period, and it's already implanted in the endometrium. In the gastrulation, the ectoderm forms the outer layer, endoderm lines the digestive tract, and mesoderm partly fills the space between endoderm and ectoderms. And after that, organogenesis occurs, and during organogenesis, various regions of germ layers develop into rudimentary organs, cells, differentiate and form tissues and organs, and factors that affect organogenesis are the genes, cells interactions, and cell movements. Okay, let me shortly explain the development of human embryo. The first period we called segmentation or cleavage period, and human pregnancy usually takes 40 weeks. In the first week, with the help of this human chorionic gonadotropin, it secretes this hormone and it maintains the corpus luteum. And corpus luteum sends progesterone and secrete progesterone and estrogen hormone, so endometrium remains and implantation occurs. But when the placenta forms after 4th or 5th week, the placenta forms from the thromboblast, and this placenta, there is some kind of blood vessels. Blood from the embryo travel to placenta through the arteries and veins of the umbilical cord. And in the umbilical cord, the veins and arteries are belong to the fetus or embryo. And in the umbilical cord, there are two, let me show you, there are 2 arteries and 1 vein in the umbilical cord, and these 2 arteries and vein is attached to the embryo, and never the embryo, the fetus blood and the mother blood never mixed, but this umbilical, normally all arteries carries the oxygen-rich blood, but in umbilical arteries, the carbon dioxide-rich blood is carried to the maternal blood pool. This maternal blood pool, embryo obtains oxygen and food from mother, from maternal arteries. Also, this fetus or embryo gives sand, carbon dioxide, and waste through the maternal veins. And this gas exchange, also food and waste exchange, occurs in the maternal pool, and the maternal pool refreshed frequently by the mother arteries and maternal arteries and maternal veins. Don't forget that umbilical arteries carry carbon dioxide-rich blood, umbilical vein carry oxygen-rich blood, and mother and embryo's blood never mix. Okay, in the embryologic period, in the 2 and 8 week to the 9th week, actually, we call this embryo, and blood from the embryo travel through the placenta, through arteries and of the umbilical cord and uterus via a umbilical vein. If there is splitting of the zygote into 2 blastomer, and if these 2 blastomer behave like a zygote and form 2 different embryo, we call these embryos identical twins. The first trimester means the first 3 months is the main period of organogenesis, and apoptosis is the process of programmed cell death that occurs multicellular organism to make morphology of the organism, to give shape to the organism. And at the beginning of the 8th week, embryo is nearly 13 millimeter and 5 gram. And you see the human fetal development in different weeks. Fetal periods start from 9th week to the 40 weeks, and all the major structures are present in the 8th week, and embryo is now called fetus. After the 9th week, we call this embryo fetus. And during the 3rd trimester, the last trimester, fetus grows and fills the space within embryologic membranes. Okay, what happens in general in the third, fourth, fifth, sixth, eighth, and ninth month? First, in the third month, more muscle tissue and bones have developed and bones become harder. Main development and external genitalia is finished. We can understand the sex of the fetus in the third month. In the fourth month, the fetus reaches the length of about 13 or 17 centimeters. In the fifth month, fetus is more active with increased muscle development. Also, fine hair develops on the head. In the sixth month, embryo eyebrows and eyelashes appear. Fetus weight about 50-100 grams, and fetal skin is almost transparent because of blood vessels. It is seen red. And in the seventh month, the fetus reaches a length of 37 centimeters. The amount of body fat rapidly increases. And the 8th month, fetus skin is red and wrinkled. Testes go to scrotum. And in the 9th month, the fetus reaches the length of 48 or 50 centimeters, and skin is smooth. All organs are developed. A complex this interplay of local regulators, hormones induced and regulate labor. The process is which called childbirth. Laboring is the childbirth. And these hormones are these are the pregnancy hormones. You see, this is the hormone chronic gonadotropin. It's very high in the first 3 months, but it decreases after the 3rd month. Estrogen and progesterone level increase all pregnancy because these estrogen and progesterone first start secreted from the corpus luteum, but after that this progesterone and estrogen secreted from the placenta in the remaining period of the pregnancy. And birth is brought by the series of strong rhythmic uterine contractions by the help of oxytocin hormone. It's secreted from posterior pituitary, but it is synthesized in hypothalamus. And first the baby is delivered and the placenta. Milk glands developed by the help of estrogen, progesterone, also prolactin hormone. Prolactin is the hormone secreted from the anterior pituitary, and it causes lactation. Lactation means the production of milk. And there is a positive feedback control mechanism. For example, the suckling of this baby gives some stimulus to the receptor in the nipples, and this receptor sends to the stimulus to the hypothalamus and pituitary, anterior and posterior pituitary. More oxytocin and more prolactin hormones are secreted. And these are the control of lactation. Hypothalamus from the posterior pituitary secrete oxytocin, and it causes the milk release. But also hypothalamus affect the anterior pituitary gland, and anterior pituitary gland secrete prolactin, and it increases the milk production. And suckling of baby causes more and more positive feedback mechanism on this lactation. And this is all about development. Thank you for your listening.